Good afternoon, everybody. My name is Abby Bronson, and I am the Senior Vice President of Research Strategy at PPMD. I'm delighted to be able to introduce this afternoon's webinar on a novel exon skipping therapy being developed by NS Pharma. Before I introduce our speakers, I want to let you know that we will have time for questions at the end of the talk. So if you have a question or need more clarity on something, submit your question in the chat box, and we'll answer as many as we have time for. And any that we can't answer during this webinar, we'll get to you by email after the webinar is done. And as you just heard, the webinar is being recorded, and you'll be able to access it at a later date. We post it on our website a couple days after the webinar. So now I'd like to introduce our speakers for today. We have two. We have Dr. Paula Clemens and Lauren Morgenroth. Dr. Paula Clemens, she's the study chair for this phase 2 study by NS Pharma. She's also the medical director for Synergy. She is a neurologist and scientist at the University of Pittsburgh. She has particular expertise in the care of adults with neuromuscular diseases, such as Duchenne and Becker, in addition to providing clinical care for patients with a broad array of neurological problems. She pursues both clinical and basic translational research focused on the development of treatments for genetic muscle diseases. She is the medical director of the Cooperative International Neuromuscular Research Group, which many of you know by the name of Synergy. She leads several multi-center studies focused on the natural history of and drug development for genetic muscle diseases. Lauren Morgenroth, she's the operations director for the clinical project management team for this NS Pharma phase 2 study. She's also the chief operating officer of TRINS. TRINS is a specialized neuromuscular CRO and is also the coordinating center for Synergy, an academic clinical trial network. Lauren has been involved with Synergy for over 12 years by running neuromuscular clinical studies, most focused on Duchenne. She is also a certified genetic counselor, as well as an adjunct instructor in human genetics at the University of Pittsburgh School of Public Health. Lauren has chaired numerous committees and working groups. She is currently a member of the PPMD Duchenne Connect Scientific Advisory Committee. And now I'll turn it over to Dr. Clemens so she can begin.
Thank you, Abby, for the introduction, and my thanks to Parent Project for the opportunity to present the NS065 trial to test a new exon 53 skipping drug. I would like to introduce the partners who are conducting this study. NS Pharma is joining with PharmaLex to sponsor the study. The Synergy network of study sites, led by the Synergy Coordinating Center at TRINS, is running the study. NS Pharma, PharmaLex, and TRINS, coordinating the Synergy study sites, are together partnering to run the study testing the safety and efficacy of a new exon 53 skipping drug. The parent company of the U.S. subsidiary NS Pharma is a pharmaceutical company in Japan whose name is Nippon Shinyaku. It is almost 100 years old, and it is an established large company which has one of its focuses on intractable and orphan disease, and on that basis are pursuing treatments for Duchenne muscular dystrophy. The NS Pharma subsidiary is their U.S. Location, and it's located in New Jersey. Nippon Shinyaku has 2 other overseas offices, and those are located in London and Beijing. Nippon Shinyaku also has 2 research laboratories that will support the clinical study. This slide shows the licensing activity of Nippon Shinyaku since the 1990s. Nippon Shinyaku has been growing by alliances with many companies in the drug development space. The focus of today's webinar is Nippon Shinyaku's drug development of exon 53 skipping for DMD, which is in the phase 2 clinical testing phase. Nippon Shinyaku also has earlier stage DMD research programs that are focused on other exons and will not be the topic of today's presentation. The exon skipping drug, or the NS065, is an antisense oligonucleotide with a morpholino backbone. It is delivered by IV infusion once weekly. The concept of exon skipping is to convert an out-of-frame dystrophin gene deletion to an in-frame deletion with a goal of generating a partly functional dystrophin protein. So in the schematic here, you see the situation of an exon 52 deletion, which is an out-of-frame deletion causing Duchenne muscular dystrophy. And by the introduction of skipping of exon 53 at the RNA level, the frame is restored to the transcript of the gene, and then this can result in a partially functional protein. Highlighted in yellow are some of the DMD deletions that can be addressed by an exon 53 skipping agent. And you can see that this is estimated to pertain to approximately 8% of those patients with DMD. Nippon Shinyaku previously conducted a phase 1 study of the exon 53 skipping agent in 10 DMD boys. This study was conducted in Japan, and it followed a dose-escalating model shown on this slide. So the first cohort was at a very low dose of 1.25 milligrams per kilogram, and 3 patients were treated in that cohort. The second course raised the dose to 5 milligrams per kilogram, and again, 3 participants were treated with that dose. And the final cohort at the highest dose of 20 milligrams per kilogram used in this study was a group of 4 participants. Each cohort was treated weekly with the exon 53 skipping agent for 12 weeks. And they had muscle biopsies performed before and after the treatment phase. There were no serious or severe safety signals in this first in-human study. Thus, the exon 53 skipping agent appeared to be safe. The doses in this study were too low to test for efficacy because the primary focus of this initial phase 1 study was safety alone. However, there was restored dystrophin expression seen in muscle biopsy tissue in the participant who received the highest dose of the drug. So one of the participants on the 20 milligrams per kilogram cohort who was of the highest weight and therefore received the highest absolute amount of drug. So this study, this phase 2 study of the exon 53 skipping drug will be conducted in the U.S. And as I said, this is a phase 2 study, and it's a dose-finding study to assess safety, tolerability, pharmacokinetics, and pharmacodynamics in boys with DMD. This slide shows you the clinical study design. There are two dose groups in this study, a low dose, which is 40 milligrams per kilogram dose, and a high dose, which is 80 milligrams per kilogram dose. The first cohort will be the 40 milligram per kilogram dose. Once the 40 milligram per kilogram dose proves safe, only then will the second cohort be treated with the 80 milligrams per kilogram dose. The study design is the same for both dose cohorts. Following screening, participants will have a muscle biopsy and then will be randomized to receive drug or placebo for the first 4 weeks of the study. That is called period 1. This will be double-blinded, so neither the participants nor the investigators will know which of the participants are on drug or placebo during this 4-week phase. Following period 1, all participants will enter into period 2, where they will be on the active drug for an additional 20 weeks. After the end of the study, at week 24, a second muscle biopsy will be taken. The participants at that time will be able to elect to either come off of the study drug or to continue receiving the study drug in a long-term extension study. At this point in the presentation, I am going to hand it over to Lauren Morgenroth to give you some more details about the study.
Thank you, Paula, for starting the presentation, and also I would like to thank Abby for the introduction, as well as Parent Project Muscular Dystrophy for hosting this webinar. I will finish the presentation by going through the study population as well as the eligibility criteria, and review also information regarding reimbursement for families. So this particular study will enroll 16 patients aged 4 to less than 10 years old with a mutation that is amendable to exon 53. The next slide here shows you the key inclusion criteria for the study, and a complete list is also available on clinicaltrials.gov. So this presents to you the high-level inclusion criteria. As we have said, this is an intervention that is focused for patients that have mutations that are amendable to skipping of exon 53. So we will, once you meet with the study doctor at the participating site and complete the informed consent, the study doctor will then work with you to review these eligibility criteria and confirm that you have the particular mutation. It will also be reviewed centrally, so it is also centrally reviewed. We will confirm that your son is between the ages of 4 to less than 10. We will also make sure that the participants are able to walk independently without assistive devices, for example, a cane. We will also make sure that the patients can complete the 3 standard time tests. We will not record a particular time or speed. We will be collecting just the able to perform the 10-meter run walk, the time to get up from the floor, and the time to climb 4 steps. We are just confirming that those 3 assessments can be performed. We are also making sure that all patients have been on a stable dose of glucocorticoid, and this could be either deflazacort or prednisone or prednisolone for at least 3 months. The key exclusion criteria are shown on this next slide here. Let me see. I don't know, Kaylin, if it, perfect, thank you. So we are making sure that patients do not have any evidence of any type of heart disease that may be already known, any sensitivity to any of the components of the study intervention, We are also making sure that patients have not been taking any other investigational drug currently or within the 3 months prior to receiving this intervention. We will also make sure that patients have not received or had a surgery within 3 months. And we will also do tests during that screening period and confirm that the patients do not have any reactions or past hepatitis B, C, or HIV. The next slide here shows you the overall study assessments. As you've heard from Dr. Clemens, this is primarily at safety. So the 4 bubbles that you see on the bottom part are regarding the safety. We will be measuring blood draws for safety labs throughout the study. We will collect vital signs at every visit. There will be frequent physical exams to, again, monitor safety, and we will also collect urine. We are also, as you heard from the study design from Dr. Clemens, collecting 2 muscle biopsies, one during the screening and one at the end of the study. We will also collect some strength and function assessments. So we will be collecting the 3 time tests that I described during the screening process, as well as the North Star ambulatory assessment, the 6-minute walk test, as well as the quantitative measurement system, where we will have a quantitative way of measuring the strength of the boys at screening, midpoint in the study, which is week 13, and at the end of the study. The sponsor is working with a company named Greenfire to help reduce the burden for costs of travel and out-of-pocket expenses with regards to traveling to these participating sites. Greenfire will help the families make travel arrangements as well as issue them a card to be able to reimburse for those out-of-pocket expenses. This next slide shows you the map of the U.S. As you heard from Dr. Clemens, this trial is being done in the U.S., and these are the current sites that are planned or anticipated to participate. Enrollment has not yet begun, but we hope to enroll the first patient at the end of this month or early December. Additional sites and additional information will be available on clinicaltrials.gov as these sites obtain the necessary approvals. Just this past week, on November 9th, the company received very good news from the Food and Drug Administration regarding being granted fast-track designation for the NS065 compound for the treatment of Duchenne muscular dystrophy. And these slides will be available on PPMD's website to click on the parent company website to read more about this press release. Finally, I want to also share with everyone the contact information for any questions for this trial. I am the primary point of contact on behalf of the sponsor to help families with regards to questions regarding the study. The study is also posted on clinicaltrials.gov, and this provides the number for that posting. And we will have time now to answer any questions from participants. This is a recent picture of the study team that met just this past weekend in Pittsburgh for the investigator meeting. So on behalf of NS Pharma and the rest of the study team, we would like to thank you and open it up for questions.
So as people type questions, and I hope you all do because we have some time for questions, I have a couple. First of all, could you talk a little bit, I mean, all the other exon skipping companies have chosen exon 51 to start with. Why did you start with exon 53?
I'll attempt to answer that. So I think it was really the timing, clearly the whole development is being driven by the most, the exon skipping agents that would hit the highest number of potential patients. And so 53 and 45 are address sort of the second largest number of groups of patients after 51.
Okay. And so you just thought maybe it was like better to start with 53, the second largest. So then we have some questions that have come in on the phase 2 study design. First of all, the dosing regimens from the phase 1 and phase 2 are different, correct?
That's correct.
Why?
The highest dose in phase 1 was 20 milligrams per kilogram, and the lowest dose in the phase 2 study will essentially dose escalate that to the next level, which is 40 milligrams per kilogram.
And then what's the high dose in the phase 2?
And the high dose is 80 milligrams per kilogram.
Okay. And I guess the question was why? Why are you, or how did you make the leap to go up to those higher doses?
So I think in a phase 1 study really looks at safety, and the indicator so far is that the intervention is safe. And when one is doing a dose escalation, one is looking for both the maximal tolerated dose as well as the minimal effective dose. And we feel that we need to continue that dose escalation to learn both of those things.
Okay. There was, again, on the phase 2 study design, there's a question about you have, after I think 24 weeks, people are allowed to choose if they want to stay on drug and go into extension up to 48 weeks. After that 48-week period, if they've chosen to be on drug, could there be an option to even extend beyond that?
Yes, I think that the 48 weeks is somewhat arbitrary, and how long how long an extension phase is will depend both on the performance of the drug, the analysis of the data from the phase 2A study, and plans for the future of the drug. Right. Okay. No, that sounds reasonable. And again, on the phase 2 design, so is the age requirement, someone asked, less than 10 at dosing or at screening?
Well, I can answer that one, Abby. That's at dosing.
Okay, at dosing. So they can be screened when they haven't, as long as they're less than 10 when they're screened, they're good to go.
Yes.
Actually, what you just said was as long as they're less than 10 when they're dosed, right, Lauren?
And they're dosed, right.
Okay, yeah. Dosed, yeah.
So they can get that dose on the 364th day of their 9th year.
Got it. And what muscle are you biopsying?
The bicep.
Okay. And you're going to alternate, or?
Yes. So the first one will be either right, left, and then the second one will be the one that was not biopsied at screening.
Okay. And there was a question about, and this comes up quite frequently, and, I don't, maybe you haven't even thought about this, but when would this drug be opened or the study be open to older patients and non-ambulatory patients? Sort of some other populations that we're always very concerned about.
So the company obviously wants to make sure this is safe in this age group, and then we'll have additional study designs in other populations should this show to be safe.
Okay. What method, is there a question about what method will be used to measure dystrophin? I know that is something that's of keen interest to our community.
Sure. That's actually been very carefully planned because clearly that's a really key factor, and a highly valuable, the muscle biopsies are an extremely valuable resource. So the plan is the analysis of expression from the muscle biopsies will be done in a contracting laboratory called AGATA. They will be measuring dystrophin quantity by Western blot and also by mass spectrometry. They will also be doing immunohistochemistry on sections for dystrophin expression, and finally they'll be measuring RNA expression levels as well.
Okay. So probably the mass spec is probably the new method that's being used in this study to quantify dystrophin. Is that correct?
That's correct. That's correct.
I mean, it's been used, but not in a regulatory sense before.
That is correct. It's it's a highly reproducible assay. It may not be quite as sensitive as Western blot, which is why they will both be done. And I just want to actually emphasize that there is a tremendous effort for this study going into making sure that the quality of the muscle biopsies all the way from collection to preparation to shipping to analysis is just at the highest level of quality. Consider that of utmost importance.
Yeah, that's critical. So there are some questions coming in, and I know we talked a little bit about it during the talk. And again, it's about the dystrophin quantification, the amount of dystrophin that was produced in the phase 1. Can you go over that again? Because I think people are still having a few questions about it.
Sure.
Was it one, you quantified dystrophin for all the patients in the phase 1?
Right. And I was not involved in doing the phase 1 study. That was done entirely in Japan. And remember that those doses were quite low.
Right.
And so the dystrophin was analyzed. My understanding is that where dystrophin expression was seen was in the participant who received the highest dose of drug. Really, with the design of the study, there was no anticipation to see dystrophin expression at all. And so really you can't look at it. This is what we call an N=1. It's really just a hint. It doesn't really tell you anything because the question was not being asked, so you can't derive the answer in a study where you're not asking the question.
Right, but clearly you got the answers you needed to move on to the phase 2.
Exactly. That's the most important message to say.
Yeah, and then also, so following on that, there's a question about, you are making a leap from 20 to 40 and 80, which are higher. So what gives you the confidence that there won't be any adverse effect or there'll be no safety issues? Do you have animal data that correlates? That supports that?
Sure. So there's been very extensive non-clinical testing done in animal models, including monkeys, and really that's where that's what gives the confidence that this is a reasonable, place to start. Anytime you're doing these kind of drug studies, there's a danger of going too high, but there's also a danger of going too low, and it's a difficult decision-making process. It involves many experts. It involves the input of the FDA. It involves lots of consultants who weigh in, but based on maybe the simple answer is that largely determined by the non-clinical studies in animal species, that this seems like a reasonable first choice or a reasonable starting point. The safety, of course, will be of paramount importance and will be very, very closely monitored throughout the study.
Okay. Actually, this sort of relates to safety, long-term safety. Are the phase 1 kids still taking the drug?
I do not have the answer to that.
You don't know.
I'm not sure, and I actually, as I saw that question, Abby, I did email the sponsor, so maybe we can get an answer before the end of the call, but I can answer the next question that shows up on the chat.
Okay. Good.
So the screening process, so the question is, how long is the screening process? The screening process will span 28 days. There will be a minimum of 2 visits, but because of scheduling with the muscle biopsy, there could be a total of 3 visits. And really, the primary activities that are happening during that screening are, first and foremost, the informed consent and meeting with the study doctor and study team to review all of the criteria for inclusion so that the families and the child understands again, the study design, the schedule of assessments, and then there will be some of the routine assessments that are done again for safety, so blood draws, physical exams, review of medical history, and then if the participant meets, then and only then would the muscle biopsy be performed. So we are making sure they meet all other criteria before the muscle biopsy will be done.
Okay. Let's see. I'm going through the questions, and we will have time for more, so if anybody has any questions, keep on writing them in. There's a question, I think we've kind of talked about this, but someone wanted to know about, converting doses in animals to people, which is hard, but have there been any adverse events, and at what kind of doses? And I think probably this has been well studied because you have to look at this as part of your IND package, but do you want to just comment on that?
Right. So what we know from the animal studies is that, the drug does distribute throughout the body. It does concentrate in the kidney. There have been no serious or severe safety signals or concerns in the non-clinical studies. But there will be extensive urine testing in this study simply because the drug does concentrate there, and so that will be an area that will be monitored closely.
Okay. And this is probably the big one, not the big one, but sort of a more abstract one, but how does this product differ from the Sarepta 53 skipping product? I mean, both skip exon 53, both are morpholinos, so how is it different?
Right. So they as you say, they both have morpholinos, which is the backbone. And remember, these are synthetic nucleotides which bind to the RNA. And so they essentially are a different sequence between the two of them. They have, as I understand it, a little different length, so that the NS Pharma agent is shorter, and so then they bind to a little different place in the RNA. But remember that the goal of both is to bind to the RNA and to interfere with the splicing of exons so that it promotes the skipping of exon 53. Right.
Okay. So basically, that sequence, that tells where the binding is going to take place within the exon. It doesn't bind the whole exon.
On the RNA, correct.
Yeah, right.
Correct.
Okay.
These are short oligonucleotides.
Right. So this probably binds to a different place within the exon, and it probably is a shorter sequence. Mm-hmm. Right. So it's a little bit different. Let's see. So do we have any other questions here to answer? We answered. We're going to get back to people on if the phase 1 kids are still taking the drug. Who is the doctor at the Chicago location, and will the Chicago site be open by the end of the year?
So the PI for the Chicago is Dr. Rao with co-PI Nancy Kuntz. We are not currently projecting that site to be open for the first patient to be enrolled by the end of the year. It'll be early 2017 based on our latest projection. But it's important if you are potentially aging out to contact the email address on the screen that is still on the webinar to make any type of arrangements to potentially go to another site that could be open earlier. So just if you do have a concern with the age, please reach out to the email address on the screen.
Okay. What is I don't see any other questions. I think someone asked to convince me. Oh, here we go. The 16 boys you're recruiting, are they all from the U.S.?
Yes.
Okay. And if the clinical trials go well, are you looking to optimize dosing, like monthly dosing or something like that?
I think that's really out in the future.
Yeah, too far out. Okay. I think that's all the questions we're going to answer today. Let me see. I mean, I think we have some questions here about, convince me this is better than Sarepta, and I think everyone has to make that choice in consultation with their healthcare provider because, that's a personal decision that you all have to evaluate based on the facts. And this is very early stage at this point, so I don't think that we can really comment on things like that. There's a question coming in. How about Canadian patients?
So for patients in Canada, right, for patients in Canada, I would encourage the families to reach out to us. We are exploring Canada. It's something we need to check with the regulations and the regulatory as well as the sponsor, but continue to contact the individuals that you've been contacting. You are speaking to the correct individuals.
Well, someone's asking what site, I think there's people asking what sites will be open by the end of the year, and is that going to be posted on clinicaltrials.gov pretty soon? Because usually they have what sites are recruiting and what sites are just, they're sites, but they're not recruiting yet.
Right. We're working to update the clinical trials this week because of the webinar. That is something we are prioritizing to have updated very soon. But again, reach out to the email address on the screen so we can make sure if you have a concern with age, we can get you to the right sites that will be open.
Okay, and someone asked about the results of the phase 1 study, and in the presentation, these slides are going to be posted to our website, and the links to all the publications that were mentioned in the presentation will be available. They'll be live, so you can just link in. And Paula and Lauren, do you know whether the phase 1 study results published? Or was it just a press release that went out?
So they were presented, and I think in the slide that's the presentation. I don't know whether they have reached the stage of being published, but I can get back to you on that.
Okay.
And Abby, we've also received the answer back from the sponsor. The 10 patients from Japan in the phase 1 are not continuing to receive it currently. They're doing now longer preclinical studies.
Okay. So again, there's questions that are coming in about why would I want to have my son in this trial versus the Sarepta trial, and I think that we've highlighted some differences between the two products today. And dosing certainly is one of those differences. But again, I just want to emphasize that you take the facts and go back. I think this is a consultative process that you have to talk with your healthcare provider about because they will be the ones together with you to be able to make the best decision for your family. Oh, and someone wants to know is there a placebo arm in this trial?
So there's just the 4-week double-blind placebo phase, at the beginning of the trial, and that's the period 1, and then that's followed with 20 weeks of everyone on active drug. So there's a placebo phase only for the first 4 weeks of each cohort.
And then everyone's rolled into drug.
And I guess it was on the slide, but I didn't say it. That randomization is a 3-to-1. So there'll be, in each cohort, there would be, during that randomized double-blind placebo-controlled 4-week phase, there will be 6 participants on drug and 2 on placebo. So at the end of the study, there'll be 16 participants, 4 of whom received placebo during the first 4 weeks of their cohort.
Okay. So someone was asking this, because Sarepta felt pressure from the FDA for the 96-week placebo, but I think it's really important to keep in mind the intent of this trial versus the trial that's being mentioned about Sarepta, because this is a phase 2. Polly, do you want to go back through or talk a little bit about what kind of results and what information you're trying to get from this trial? Because I think that will explain why someone's asking about, such short placebo time.
Right. So actually, the design for this was developed in conversation with the FDA during a pre-IND meeting, and their main concern was that there be a placebo phase for initial acute safety, and that is the reason for having the 4 weeks double-blind placebo-controlled phase. There are different and, of course, evolving strategies for reaching the point of showing that a drug is effective and gaining approval. And that probably gets into more than I can go into at this time. But suffice it to say that, this is a very organic process that all study groups are engaged in.
Okay, well, I think that is all the questions that we have on the chat. And as I said before, if you think of anything as you go back through this presentation or have any more questions, you can email me. You can email Lauren. There's an email address right on the screen right now. And as Lauren said, they're working with clinicaltrials.gov, so that information will be updated soon. I really want to thank the speakers today for taking the time to update us all on this really exciting trial. And if there's nothing else, I think we can close.
Thank you very much.
Thank you.
All right.